everyone, and thank you for coming to my poster entitled Role of GWAS Candidate Gene SCYL3 and TDP-43 Expression and Aggregation. The TDP-43 is a multifactorial protein that binds DNA and RNA and plays several different roles in RNA alternative splicing, metabolism, trafficking, and mRNA expression. It has been implicated in a range of neurodegenerative diseases, also known as TDP-43 proteinopathies. The main pathological hallmarks of these diseases are cytoplasmic inclusions that consist of TDP-43. These cytoplasmic inclusions can be found in approximately 97% of ALS cases, 45% of FTD cases, and 57% of AD cases. In addition, TDP pathology is found with increasing frequency in the brains of elderly individuals defined as 80 years or older, a condition recently named Limbic-predominant age-related TDP-43 Encephalopathy, or LATE for short. This late pathology is also seen in 50 to 60% of brains with Alzheimer's disease. Other TDP-43 pathological hallmarks occur when TDP-43 becomes depleted from the nucleus, where it is normally located. This depletion causes RNA processing defects and destabilizes several RNA transcripts. As TDP-43 becomes depleted in the nucleus, it can accumulate in the cytoplasm, where it forms aggregates through liquid-liquid phase separation, where it can affect autophagy and mitochondrial dysfunction, and has been shown to play a role in stress granule dynamics. In order to learn more about the genetic landscape of TDP-43 proteinopathies, we performed a genome-wide association study, or GWAS for short, to look at genetic modifiers that specifically modulate TDP-43 pathology and severity. They performed sequencing on a post-mortem cohort of 207 patients with moderate to severe AD cases with variable presence of concurrent late pathology, and found a genome-wide significant signal on chromosome 1 that is mapped to an intron in a gene called SCYL3. Having 1 or 2 copies of the risk allele A was associated with a more severe TDP-43 pathology stage. So what exactly is SCYL3? SCYL3 stands for SCY1-like pseudokinase 3 and is a protein coding gene. It is part of the SCYL family, which consists of SCYL1, SCYL2, and SCYL3, and has been implicated in neuronal function and development. However, the exact biological function of SCYL3 remains unknown. It has been proposed by previous studies that SCYL3 may play a role in protein phosphorylation, cell migration, spinal cord motor neuron differentiation, cellular protein localization, and neuron development. These biological roles make SCYL3 a promising gene candidate for neurodegenerative diseases, and the reason that our lab decided to further investigate how SCYL3 may be related to TDP-43 endogenous functions and pathology. Here's an image that shows how SCYL3 co-localizes with Golgi functions like Golgi, shown in green, and is found predominantly in the Golgi apparatus due to myristylation. In order to test how SCYL3 may be interacting or affecting TDP-43 endogenous functions, we performed a siRNA knockdown of SCYL3 and looked at the effects on TDP-43 expression. Here we show in our qPCR results that we were able to achieve a robust knockdown of SCYL3. Interestingly, we also saw that TDP-43 expression was also reduced by approximately 30%. Our immunofluorescent images also demonstrate how knockdown of SCYL3 causes a reduction in TDP-43 intensity within the nucleus. We then wanted to test how SCYL3 knockdown affects not only TDP-43 endogenous function, but TDP-43 pathology. We used a novel optogenetics method developed by a previous PhD student in our lab who created a construct called opto-TDP-43. Essentially, the Cry2 protein clusters and aggregates when exposed to blue light. This is normally reversible when Cry2 is no longer exposed to the light. However, when TDP-43 and a fluorescence reporter is connected to Cry2, we see these aggregates are irreversible after exposure to the light. Using this opto-TDP-43 construct as a basis, I created several monoclonal stable cell lines with a TET3G reporter that is inducible with doxycycline. Using this stable cell line, I treated them with SCYL3 siRNA for 72 hours, induced the cells for 48 hours with oxy, and exposed the cells to blue light for 5 hours prior to fixation for IF. Interestingly, our data shows that when we knock down SCYL3 in the opto-TDP cell line and induce TDP-43 aggregates with light, Knockdown of SCYL3 resulted in a higher number of puncta on average per cell as compared to our control group. There is no significant difference in puncta size, however. Conversely, when we overexpressed either an empty GFP vector or a SCYL3 GFP construct in our opto-TDP43 line, we saw that there was a reduction in puncta count per cell and a decrease in the puncta size when SCYL3 is overexpressed compared to the empty GFP vector. And so to summarize, we are running functional analysis of SCYL3 based on a GWAS study performed by Dr. Koffler. siRNA knockdown of SCYL3 shows that there is a loss of nuclear TDP43 levels. However, it does not appear that mislocalization of TDP43 occurs under these conditions. Furthermore, using our opto-TDP43 cellular model that mimics the pathology, we were able to demonstrate that loss of SCYL3 results in an increase of cytoplasmic TDP43 puncta and increase of severity compared to the control siRNA. Conversely, overexpressing SCYL3 results in the opposite effect, where there is a decrease of cytoplasmic TDP43 puncta count and average size compared to the empty GFP vector control. These findings suggest that SCYL3 modulates cellular expression levels of TDP43 and may act as a protective factor against formation of pathologic cytoplasmic TDP43 aggregates. Further directions include sequencing patient cell lines for the risk genotype and creating iPSC cell lines from these patient samples to differentiate into motor neurons for further characterization. In addition, we've hypothesized that SCYL3 may be affecting protein turnover, perhaps through upstream pathways that eventually lead to TDP43 aberrations. We plan on performing RNA sequencing to look at changes in global expression to see what known transcripts related to TDP43 pathology are also affected by knockdown of SCYL3 to tease out other components of the pathway SCYL3 may be affecting. Finally, I would like to acknowledge my lab, the Donnelly Lab, our collaborators, Dr. Julia Koffler, Dr. Ken Sen Fan, and Dr. Ilias Cambo, and the numerous funding sources for their help, support, and guidance in performing this research. Thank you.